In my talk at ICRISAT today, I'm going to be covering all aspects of our programme, which represents the last 10 years of pre-breeding research at NIAB. In this time, we've been working extensively on generation of new genetic material to understand complex traits in wheat and to deliver this information to private sector breeders and to researchers throughout the UK and the world. This material includes precise genetic stocks for key adaptation genes like photoperiod response, in which we move allelic variants into common genetic backgrounds and assess their effects in the field in the UK, and then extend them out throughout Europe to understand more about wheat's adaptation to the environment. We also look at new genetic diversity and how it can be deployed in breeding. To do this, we resynthesize the hexaploid genome of wheat by going back to its ancestral species. And these include a range of Triticum dicocoides and dicoccums, so wild and domesticated emmer wheats, which represent the A and the B tetraploid genomes of wheat, as well as to goat grass, which more recently hybridized with tetraploid donors, giving rise to what we now know as bred wheat. And these goat grass accessions occur over a wide genetic range and have extensive geographic, functional, and genetic diversity. So we're bringing those into our wheat pre-breeding program in order to extend the range of diversity that's available for breeders to improve wheat production in the UK and worldwide. And we make all of our material publicly available and IP free, so it's completely available for people to use either in research or in breeding throughout the world. As well as looking at new sources of genetic diversity, we also look at highly recombined populations, so looking at new combinations of alleles and new genetic combinations and what these can give us in terms of resistance to diseases and improved agronomic traits. So we use multi-founder populations called magic populations to dissect the genetic control of complex traits and to extract individual lines which combine favourable alleles for advantageous performance. And we take these from the program and we generate additional pre-breeding lines, as well as distributing them to partners in academia and in industry. We also work extensively on quantitative genetics, applying techniques such as genome-wide association mapping and genomic selection for the improvement of quantitative trait performance in wheat.